Assalamu alaikum. I'm Gulab. My registration number is MLT12019 and 2025. Today, my presentation topic is test for differentiation of Streptococci bacteria. So, I will discuss about different tests used for the differentiation of Streptococci bacteria in the next coming slides. Test for differentiation of Streptococci. Differentiating between various species or groups of Streptococci is important for clinical diagnosis and treatment. Clinical diagnosis and treatment are very important for the differentiation between different species of the Streptococci, and it is very important for differentiating of different groups of the Streptococci bacteria. Now, here are some common tests used for the differentiation of Streptococci. In the next coming slides, we will discuss about the different tests which are performed, which are done for the Streptococci differentiations. Gram staining. Purposes of gram staining is initial classification based on cell wall characteristics. In this classification, we will see that whether the cell wall of the bacteria is made up of what type of materials. And the next point, distinguish between gram-positive or gram-negative bacteria. Whether the bacteria is gram-positive or gram-negative. In gram staining, we will know about the bacteria, that whether this bacteria is a gram-positive bacteria or a gram-negative bacteria. In the gram staining, gram-positive bacteria show blue or purple color after gram staining in laboratory test. Whenever we perform gram staining test in the laboratory test, gram-positive bacteria will show blue color. And also they have thick cell walls. Gram-positive bacteria have thick cell wall. Gram-negative bacteria show pink or red color on staining and have thin walls. Whenever we perform gram staining for the gram-negative bacteria, it will show pink color or red color. And also gram-negative bacteria have a thin cell wall. Gram-negative and gram-positive bacteria excrete different toxins and affect the body in different ways. In this slide, we will discuss about the hemolysis pattern on blood agar. Purpose of the hemolysis pattern on blood agar is identifying and detecting the ability of Streptococci bacteria to lyse or split the red blood cells. That how much Streptococci will lyse or break how much blood cells. Clear zones around colonies complete lysis. Beta is lysis of RBCs and media looks completely transparent around the colonies. Alpha hemolysis is the greenish zone around the colonies, or it is a partial lysis. It is caused by the damage but not lysis of RBCs in the blood. In the alpha hemolysis, the media is translucent with the greenish things around the colonies. The next one is gamma hemolysis. In the gamma hemolysis, there is no hemolysis. It means that blood is not lysed, or you can say divided in the gamma hemolysis. In this slide, we will discuss about the catalase test. Purposes for the catalase test is distinguishing between catalase negative streptococci and catalase positive staphylococci. It means that in the catalase test, we will differentiate between whether the bacteria is catalase negative streptococci or catalase positive staphylococci. So, what is the catalase negative streptococci, or you can say what is catalase negative? Catalase negative are the facultative anaerobes that only ferment and do not respire using oxygen as a terminal electron acceptor. Here are some examples of catalase negative bacteria. Enterococci, streptococci, and lactococci are the example of catalase negative bacteria. Now we will discuss about the catalase positive bacteria. Catalase positive bacteria include the strict aerobes as well as facultative anaerobes, although they all have the ability to respire using oxygen as a terminal electron acceptor. Here are some examples of catalase positive bacteria. Candida, E. coli, and Staphylococci are the example of catalase positive bacteria. In this slide, we will discuss about the bacillation sensitivity test. Purposes for the bacillation sensitivity test are differentiating between beta hemolysis streptococci bacteria. It means that bacillation sensitivity test is a differential test used to distinguish between the organisms sensitive to antibiotic bacillation and those which are not. Group A streptococci are sensitive to bacillation, while other groups are resident. Now, what is bacillation? Bacillation is a peptide antibiotic produced by the Bacillus subtilis. It plays an important role in the inhibition of cell wall synthesis and disrupt the cell membrane. In this slide, we will discuss about the aptokine sensitivity test. Purposes for the aptokine sensitivity test is differentiating between alpha hemolytic streptococci bacteria. Streptococcus pneumonia is sensitive to aptokine, while other alpha hemolytic streptococci are not sensitive to the aptokine sensitivity. In this slide, we will discuss about the bile esculin test. Purposes for the bile esculin test is identification of group D streptococci, whether the group D streptococci is present or not. Enterococci and some non-enterococci can hydrolyze esculin in the presence of bile. In this slide, we will discuss about the PYR test. Purposes for the PYR test is identification of beta hemolytic streptococci bacteria, which detects the presence of PYR enzymes. In this slide, we will discuss about the serological typing. Purposes for the serological typing is identifying specific serotypes within certain streptococcal groups, which means that which utilize specific antibodies to differentiate strains. Here are some molecular techniques used like PCR. Purposes of PCR is detection of specific DNA sequences for species identification. PCR are used for species identification. PCR says for identifying streptococcal species or virulence genes. In this slide, we will discuss about the antibiotic susceptibility testing. Purposes of the antibiotic susceptibility testing is assigning the susceptibility of streptococci to various antibiotics. which helps guide appropriate antibiotic therapy. In this slide, we will discuss about the hepuride hydrolyze test. Purpose of the hepuride hydrolyze test is differentiating of group B Streptococci bacteria. Group B Streptococci hydrolyze hepuride. These tests, when used in combination, allow the accurate identification of differentiation of streptococcal species, aiding clinician in choosing appropriate treatment and understanding the potential virulence of the bacteria involved. Thank you so much for watching us.